Hello, everyone. So, for this discussion, we will be discussing chapter 25, Lipid Metabolism. So, my reference here is Stephen Stoker instead of Bettelheim because Bettelheim book has combined both the carbohydrates, lipids, and protein metabolism. So, for this one, let's just discuss the following for lipid metabolism. So, lipid digestion, no, is dietary lipids first contains 98% of triacylglycerol. So if you can remember on our lipid discussion, we have triglyceride, triacylglycerol, or tags, which includes fats and oils. No, so salivary enzyme in the mouth has no effect on lipids. That's why the digestion of your lipid starts with your intestines, no, not in your mouth. But the digestion of your carbohydrates starts with your mouth because of the salivary amylase that we have, as well as the pancreatic amylase, which is released in the small intestine. So for this one, tags doesn't start their metabolism on your mouth. So which tags are water insoluble, as you can remember. And these salivary enzymes usually are water soluble. So in the stomach, most but not all, tags change physically into small globules or droplets. We call it chyme. So chyme, which floats above the other materials. And it is physically not chemical process. It just splits the huge droplets of your fats into smaller one, just like when you are using hand soap in oil whenever you're cleaning your pan with an oil with it. Now you'll notice that the texture and the clarity of your oil, of your clear oil, becomes blurry. It's not becoming blurry, but instead, no, your huge globules or the huge globules of your oils are being splitted into tiny droplets. That's why it appears blurry. So that same process is just physical. There's no transformation of one form to another. It's just physical. It's just splitting the huge globules into smaller droplets for easy digestion. So that is what is happening in your stomachs. Okay? In your stomach, sorry. Okay, so it starts with stomach. There's a gastric lipase enzyme that hydrolyzes your triacylglycerol ester bonds, and about 10% of your tags are hydrolyzed here. So high fat foods stay in the stomach for a longer time, and high fat meals causes a feeling of being full for a longer period of time. So remember that before you eat a high amount of fat or very fatty food. So we have your glycerol. If you can remember this structure from your lipid metabolism, we have the triacylglycerol, which is a glycerol with, from its name, triacylglycerol, meaning there's a three fatty acid attached to your glycerol, and this fatty acid can be long chains of carbon to carbon compound, which makes it a non-soluble, non-water soluble compound. So if you add water with it, no, from the looks of it, no, without this enzyme, this water and triacylglycerol will not create a reaction. But we have this lipase that helps on breaking down of your huge fat globules into smaller one, and then it will be um and then the other fatty acid will be removed. So it become monoacylglycerol, or a simple globule of fats that is easier to digest when it arrives at your intestine. So I also have that free fatty acid that can be utilized by our body on other functions that needed fatty acids. Okay, so this chyme enters into a small intestine and is emulsified. No, so emulsification again is the process of splitting huge globules, similar to what the gastric lipase have done to you to the huge globules of your fatty foods. No, so on this end, no, the emulsifying agent is your bile salt, meaning on my example just a while ago, when you clean your pan full of oils, you add a soap on it, no, you'll notice becomes blurry. So what happened there is our term called emulsification. No, on that example, our soap is our bile salt, no? So pancreatic lipase hydrolyze. By the way, pancreatic lipase no, is produced on, of course, on your pancreas, and they are being released, no, there's a connection from your pancreas to your small intestine, and this pancreatic lipase is released there so that it will aid in the hydrolysis, no, or the hydrolysis of your ester bond linkages of your fatty acid units and glycerol. So fatty acid, monoacyl glycerol, and bile salts combine into small droplets we call micelles, no? And small enough to be absorbed through the intestinal cell membrane and absorbed through the bloodstream. So yung lipid digestion natin continues. In the intestinal cells, monoacylglycerol and free fatty acids are repackaged to form your tags. No? Pag nandun na sila sa mga cells, these new tags combined with membrane lipids like phospholipids and cholesterol and water-soluble protein to form chylomicron. So what is this chylomicron? So what happened really is from your small intestine, they are broken down. Starting from your stomach to your small intestine, the huge globules are broken down. From there, no, it is being absorbed, broken down so that it can be absorbed by your smaller cells. Kapag nasa loob na yung mga molecules na yon, kapag nasa loob na ng cell, magre-repackage sila to form a tag again or triacylglycerol. In that case, it is combined not just with tags but also on the membrane lipids now we call um, uh, sorry, these tags, no, combined with the lipids and water-soluble proteins, ang tawag na natin dyan is chylomicrons. Okay? Chylomicrons is what we call lipoprotein, meaning they are a combination of your lipids and proteins. So this chylomicron, they transported these tags, no, these tags from intestinal cell into the lymphatic system and the bloodstream. So aside from chylomicrons, we have other lipoproteins such as your HDL and your LDL, no? And there's four of them, the lipoproteins, and you may remember HDL is the high density lipoprotein. No, they are the healthy proteins, and they are good, no, if they are higher than your LDL. But of course, it is still carrying a lot of fat, so too much of it is not also good. LDL is your, kumbaga, bad cholesterol. So we do not want to see LDL high in your blood test, no? Another one is your VLDL, which is further converted into LDL throughout the process. But let's focus first on your chylomicrons, okay? Chylomicrons is your lipoproteins that transports your triacylglycerol from your intestinal cells through your lymphatic system and your bloodstream. So in the bloodstream, your triacylglycerol are completely hydrolyzed by lipase enzyme. So fatty acid and glycerol are absorbed by the cells and are either broken down to acetyl-CoA for energy. As you can remember, we have used this, no, in the production of your energy on our discussion of energetics. So some of the fatty acid and glycerol are converted to acetyl-CoA for energy and repacked as stored in lipids. So during digestion, triacylglycerol are converted by lipases to blank and blank. The answer for this letter C, monoacylglycerol and free fatty acid. Okay. Next is the adipose tissue. So this is the storage of your fats, right? So ito yung taba, no? Most cells have limited capability for tag storage. So tags are stored in specialized cells we call adipocytes, no? Ito yung cells ng mga taba natin sa katawan, no? Na naimpok na mga tag. Okay, so they are found in adipose tissue, and adipose tissue is the largest cell in the body where the cytoplasm is replaced with large TAG or triacylglycerol droplets. If you're going to check this adipocytes under the microscope, you will notice that they look like a blob or they look like a fat globule with a nucleus. So they are located primarily beneath the skin. Okay, let me look for a photo of that adipocyte so you may notice them once you've seen them, especially in subject like histology or anatomy physiology. Okay, so this is what an adipose tissue looks like. So instead of seeing the parts or the intracellular or the organelles, I'm sorry. Instead of seeing the organelles, you'll notice that there's a huge bleb, no? It looks like the main cytoplasm of this adipocyte is already replaced with your tag or your fats. Okay, so this is a white adipocyte, beige and brown adipocyte. So you'll notice that there's a nucleus, no, on its peripheral na, because they are replaced already with your um, fats or your tags. Okay, let me know if you didn't see that photos, but let's go back to this discussion. Hold on. So having adipose tissue is not bad, no? Since adipose tissue has a lot of function that is needed by our body. Say, for example, it acts as an insulator to our body against heat loss, and it can also protect us from physical shock. So this is also a vital part of our body. So this is a structure characterized by your adipose tissue. This is the main cytoplasm. We have the nucleus. The cytoplasm is already replaced with this large central globule of your triacylglycerol. So we also have the hydrolysis of your tags, which involves several hormones trigger the hydrolysis. These are the CAMP, no? It activates the hormone sensitive lipase or HSL, and it releases a glycerol and fatty acid into the bloodstream after a triacylglycerol mobilization. So on an average, there's 10% of TAG replaced every day. Okay, so tags and energy reserves. Triacylglycerol, energy reserves or fat reserves are human body's major source of stored energy. Okay, so energy reserves associated with protein, glycogen, and glucose are small to very small when compared to fat research. So you can pause this video if you wanted to answer this one. I'm gonna show you the answer after this. Okay, the answer for this one is cyclic AMP. Okay. Okay, so we'll move forward glycerol metabolism. So after entering the bloodstream, they are taken to the liver or kidney and converted to dihydroxyacetone. If you can remember this one, this is present in our glycolysis dihydroxyacetone phosphate. And there are two ways, no? We have phosphorylation of primary hydroxyl group of the glycerol. We have the oxidation of your secondary alcohol groups of glycerol to a ketone. So here's your glycerol, okay? And with the use of an energy or ATP with an enzyme glycerol kinase, it will be converted to glycerol 3 phosphate, and with the use of your NAD and an enzyme glycerol 3-phosphate dehydrogenase, it can form the hydroxyketone phosphate, which can be used, no, in your carbohydrate metabolism. So again, you can pause for this video if you wanted to answer this one. Okay. The correct answer for this one is okay, letter C. Okay, now we move on to our breakdown of fatty acid to produce energy. So this process is divided into three parts. We have fatty acid must be activated by binding to CoA, coenzyme A. And number two, we have fatty acid must be transported to the mitochondrial matrix. Number three, we have fatty acid must be repeated, fatty acid spiral, oxidized to produce acetyl-CoA, FADH2, and NADH. So fatty acid activation takes place in our outer mitochondrial membrane. Fatty acids react with CoA in the presence of ATP to produce high energy acetyl-CoA, and ATP is converted into AMP. So meaning there are two phosphate release, no? Um, in this process. So here's your fatty acid. This is combined with your acyl CoA, high energy acyl CoA, and through your enzyme acyl CoA synthase, and ATP will be converted into AMP, releasing two phosphate group. Okay, the final product would be acyl CoA. Okay, fatty acid transport. This is shuttle mechanism is involved in the transport of your acyl CoA from mitochondrial membrane to mitochondrial matrix. Okay. Okay. So there's a reaction of your beta oxidation pathway. We have four reaction repeated cleave to carbon unit from carboxyl end of an acyl CoA. This process is called beta oxidation pathways. Remember that because the second carbon or beta carbon from the carboxyl end of the chain is oxidized. Okay. Through this process, FADH2 and NADH are produced. Okay. So let me take you to this photo. Hold on, if I can extract this one. Okay, so I hope you're seeing this one. Ayan. So we have four steps in beta oxidation pathway. This is your beta oxidation pathway. So the step one is dehydrogenation, meaning you're removing hydrogen. Dehyd Dehydrogenation, yan. You're dehydrating. You're removing hydrogen on it. No? On step one, you may notice that we have removed one hydrogen right here. Hold on. Okay, we have removed one hydrogen right here. Oh, sorry. I'll delete that. That's too big. Okay, so we remove one hydrogen here and another one here. That's why it's dehydrogenation. And we use FAD and FAD we use FAD, no? We use FAD and make it reduced, no? Which is naging FAD-H2 siya. So the main product here is your acetyl-CoA. Okay? We dehydrogenate acetyl-CoA to produce your FAD, no? And sorry, we dehydrogenate acyl-CoA to produce trans-enoyl CoA. So this is with the help of your FAD, and it becomes reduced FAD H2. Okay, and then the second step here is hydration. We have hydration, which is a molecule of water is added naman. So after we dehydrogenate, no, we add a molecule of water. So let's see, by adding water to this double bond right here, no, yung one bond, the first bond here will connect to an OH, and the second bond right here, since there are two free bonds connected to carbon and carbon, the other one is added one hydrogen. So that makes sense because OH plus the one hydrogen added is your H2O. Okay. The other one, the next step from here is second dehydrogenation. So meaning we again remove hydrogen from this reaction. So which hydrogen has been removed? The hydrogen that is removed is the OH, no, the OH right here. That's why it will become double bond oxygen. So this time it formed beta ketoacyl CoA. So if you can see, we use FAD right here, and it becomes FAD H2. On your second dehydration, we've used NAD and it become NADH, or reduce NAD. Okay. On your fourth step, we have thiolysis. From its name, thio, no, it means adding sulfhydryl group. Now, which part would you add is sulfhydryl group? So as you may see, thiolysis, no, instead of CoA right here, this part has been cut down. Okay. This part has been cut down. Hold on. Okay, this part has been cut down. There's a substitution, no, in the process. And your CoA and sulfur will replace this reaction. Now, since you've cut them down, this part is created. No? So this one is another new acyl CoA. You form two types of acyl CoA through this four steps process. So each loop of the pathway represents the repetition of one to four. So from repeating and repeating continuously, no, it forms different acetyl CoA groups. No, so it is a continuous process. So that's your beta oxidation of your fatty acid. Okay, so this is it. Let me stop sharing the photo. Okay. Okay, next is the unsaturated. Hold on. Okay, next is unsaturated fatty acid. So oxidation of unsaturated fatty acid, when if you can remember, unsaturated means it has double bonds or triple bonds, no? The base if you can remember the PUFAS and the PUFAS and the MUFAS on your lipid discussion differentiates your unsaturated from saturated with a double bond of unsaturated and single bond of your saturated. So it requires two additional steps compared to saturated fatty acids. We have epimerase, no, an enzyme that changes D configuration into L. D is dextrorotatory and L is levorotatory. If you can remember that in your organic chemistry, on your, I think that's chirality topic. And we have cis and trans isomerase, no, that produces trans 2,3 double bond from cis 3,4 double bond. So this again is a question. Okay, you can pause the video, but I'll show you the correct answer instantly so you will not wait for long minutes before to know the answer for this one. Okay, the answer is letter A. Okay, so for ATP production from fatty acid oxidation, fatty acid versus glucose oxidation in comparison, we have fatty acid oxidation produces a net of 120 ATP molecule by oxidation of C18 atom or carbon 18 atom or stearic acid. Two ATP molecules are needed for activation of your fatty acid. So the net ATP production is 120 molecules. One stearic acid molecule, which is C18 atom, produces 122 molecules of ATP. So the computation is below. Okay, so stoichiometric comparison, no, stearic acid produces 2.5 times more energy than glucose. Okay, so yung lipids food natin ay nagki-create ng mas maraming energy, meaning mas mataas ang calories ng ating mga fatty foods in comparison with our carbohydrates or glucose-based foods. So what is the net ATP production for 16 carbon fatty acids? So the correct answer for this one is 106. So they use, I think, a stoichiometric computation on this one. Okay, so for ketone bodies and ketogenesis, we have acetyl-CoA formed from beta oxidation pathway, just like a while ago, is processed by a citric acid cycle. So if you can remember, we use as the acetyl-CoA in our citric acid cycle, no, and the adequate balance in carbohydrate and lipid metabolism is required. So lipid carbohydrate metabolism can be disturbed by the following conditions. So dietary intake is high in fat and low in carbs, diabetic condition wherein the body cannot use glucose properly, and of course, prolonged fasting condition. So ketone bodies under low supply of oxaloacetate, the acetyl-CoA will be in excess. As a consequence, the excess acetyl-CoA is converted into a ketone body. No, we have this process we call ketogenesis. No, it involves the synthesis of our ketone bodies from acetyl-CoA. Too much ketone in our body. No, they said I think I have read one study that shows that ketone helps in our memory. So I forgot the name of that research anyway, but ketogenesis is the process or synthesis of our ketone bodies. And usually, when we starve ourselves, no, our body produces or our cell produces ketone bodies. Primary site for this process is in the liver, mitochondria, and three ketone bodies produced in our body. There are three major, which you will be discussing also in your AUBF or analysis of body fluids. We have acetoacetate, we have beta hydroxybutyrate, and we have your acetone. Okay. Okay, I think this is very important because in board exam, I remember this question. There are percentage of this that is abundant in the body, and that is the question in our boards. So there are steps involved in ketogenesis. We have condensation, second condensation, and chain cleavage, and also hydrogenation. So this is the process of your ketogenesis, no? Your C2 or carbon 2, we have the acetyl-CoA plus acetyl-CoA. It will be combined, no, through a process of condensation. It will be condensed with an enzyme CoASH, no, and then it will become or converted into acetoacetyl-CoA or C4. Now will undergo second condensation, which then converts acetoacetyl-CoA into 3-hydroxy 3-methyl-glutaryl-CoA with the use of an enzyme acetyl-CoA and H2O or water and an addition of another carbon or two carbon groups. Okay, this will then become CoA and SH and your H or hydrogen. Okay, this time since we added two carbon group from your four carbon group, it will become six carbon group. Okay, now we'll produce or we'll proceed with our chain cleavages. Now, with the use of an enzyme acetyl CoA, no, we cleave, no, we remove what two carbons, and from six carbon atom, it would become again four carbon atom. No, this one is your acetoacetate. So this acetoacetate will be consistently become four carbon atom, but will then undergo hydrogenation, in which no we uses NADH or a reduced NAD, and it will become an NAD. So it will be beta hydroxybutyrate as an end product of your 2-acetyl CoA molecule. Okay, so you can answer this question again. Okay, so the correct answer for this one is, okay, letter C. Okay, so lipogenesis versus fatty acid degradation. We have lipogenesis and we have degradation of your fatty acid. So it occurs in cell cytosol, while the degradation of fatty acid occurs in mitochondria. Okay, and the rest are its differences. Okay, so next we have our citrate malate shuttle system. So on this one, no, medyo similar siya kay citric acid cycle, in which acetyl-CoA is the starting material for lipogenesis. So acetyl-CoA needed for lipogenesis is generated in the mitochondria and must first be transported into the cytosol. We have citrate malate transport system that helps in transport acetyl-CoA to a cytosol indirectly. So this is the main citrate malate system. Okay, from acetyl on the mitochondria, we have oxaloacetate similar to citric acid cycle, and then we uses one acetyl CoA, converts it to CoASH. It would become citrate, no, would undergo a channel and a double or a semi-permeable layer of the cell, and it would still be a citrate, no. On this part, we uses CoASH and converts it back again into a acetyl CoA. So nag-return lang yung ginamit ninyo on your mitochondrial layer. Now we are on the cytosol, no, but we have to acquire again an acetyl-CoA, just like the ones that happened in your mitochondria. But this time, no, we use as ATP to convert CoASH to your acetyl-CoA. So this is the process we call lipogenesis. No, we form now an oxaloacetate again, but this time we are in the cytosol layer. Okay, after this one, we'll form a malate, and this malate will undergo into a channel, no, or a malate transporter, and it would then come back again into the mitochondrial matrix. Okay? Okay. We have the ACP complex formation, wherein all intermediate and fatty acid syntheses are linked to a carrier protein we call ACPSH. These are the giant CoA molecule. Okay? Okay, so we have chain elongation, and hold on, let me check. Okay, I think we will not be involving this in our exam, but these are important. In our chain elongation, we have four different steps. We have condensation, hydrogenation, dehydration, and hydrogenation again. Okay. Okay, this is a question, so you can jump right back, right in these questions. Okay. Now we have cholesterol, which is a necessary component of your cell membrane, no? Cholesterol has a lot of use, no? These are precursors of our bile salts, sex hormones, and adrenal hormones. So, this starts the cholesterol fate of the fatty acid generated acetyl-CoA has several steps. So, starting with C2, okay, or two carbon atom, acetyl-CoA would become mevalonate, which is a six carbon atom, as you may see. It undergoes another steps again. It will become five carbon atom, and then 30 carbon atom, and then continues to be 30 carbon atom, and then it would become your 27 carbon atom, which is then your final end product, cholesterol. Okay, so biosynthesis of cholesterol occurs, of course, in the liver also. So as an overview, acetyl-CoA is a primary link between lipid and carbohydrate. That's why there's a citric acid cycle, no, in which we utilize our acetyl-CoA right there. So we can use or utilize acetyl-CoA from the lipids to make it or to use, no, to use it as an energy. Okay. Okay, so these are the modified B vitamins function as coenzyme in lipid metabolism. B vitamins that participates in various pathways. Again, this has been discussed in carbohydrate. We have niacin, riboflavin, pantothenic, and biotin. Unlike the six B vitamins in carbohydrates, we use as four B vitamins here on our lipids. Okay. Okay, these are concept question. I'm going to show you a hold on, a connection from okay, the catabolic pathways of carbohydrates and lipids. So it's not a discussion. I just wanted to show you how they are all connected. Okay. I hope you are seeing this screen, but these are the carbohydrates metabolism. So that's why this one is a PowerPoint from Bettelheim. So I didn't use this because they are already converged into one pathway. I wanted to have a separate discussion so you can further understand carbohydrates, lipids, and proteins. On this one, this is a very good illustration in which carbohydrates undergoes glycolysis to form an ATP, but it can also undergoes and proceed into a pyruvate in which pyruvate will be converted into acetyl-CoA, and it would undergo a citric acid cycle to produce further ATP, which produce B vitamins um related molecules, no, and it will continue to have or create more ATP in oxidative phosphorylation, including the electron transport chain. So the end product of this carbohydrate, of course, is ATP. Okay, but in glycolysis kasi we do not generate much ATP, no, in here. That's why it has to undergo into this process until it reaches the citric acid cycle and oxidative to multiply the energy production in this pathway. If we are going to use lipids, no, we can proceed to digest it into glycerol, and that glycerol will be used through gluconeogenesis and undergoes glycolysis, and then again it will undergo the citric acid cycle down to the path of oxidative phosphorylation and finally ATP. Now lipids can undergo into a the fatty acids in lipid can undergo a beta oxidation, and it will enter directly the acetyl-CoA pathway. Now, from acetyl-CoA, it will then produce a citric acid cycle. Now, on our proteins, which we will discuss up next, now there are a lot of pathways. It can undergo into glucogenic amino acid and then pursue the pyruvate pathway. And then it can also undergo ketogenic amino acid, in which it can go to acetyl-CoA. It can also pursue the path of citric acid directly. Okay. So on our glucogenic amino acid, it can also undergoes an oxidative deamination in which it will become an ammonia. And this ammonia, through the process and help of the bacteria in your intestines, it would undergo a urea cycle to produce urea, which is excreted in our body through our urines. So urea is in the form of your BUN or blood urea nitrogen, which is a substance measurable in our bloodstream to assess your kidney together with your creatinine. So this one is a very helpful pathway. I think this one also is a very short discussion of all the pathways there is, no, in our both lipids, carbohydrates, and proteins. So I will not use this. I'll just use this as an overview. So on our next discussion, we will be discussing protein metabolism. Okay, so that's it for our lipid metabolism. I hope you've learned. That's it. Thank you.